Hello, this is Dr. Hanaa Assil, and these are some review questions on paper 6 of the IGCSE Combined Science. So let's take a look at some of the questions. One of the questions that we get is to plan an investigation. So this, for example, is an easy one where he says plan an investigation to compare the reducing sugar content of two different brands of apple juice. In your answer, you should include how you will determine which brand contains the most reducing sugar and how to make a fair comparison. So, this is how you test for reducing sugar. But if we're comparing different brands, then I will tell him, for example, put something, a certain volume, 20 centimeter cubed of each juice into separate test tubes. Add equal volume, so you're mentioning that you're adding equal volume of juice and equal volumes of Benedict solution to each test tube. Heat in hot water bath for the same time, so for 5 minutes, for example. And then he wants to know which has more sugar. You should know that the one that gives reddish brown precipitate has the most reducing sugar. The one that gives green or yellow has less reducing sugar, and the one that has orange has a moderate amount of reducing sugar. Okay, another type of question is where you're required to explain how to prepare a salt. For example, the student reacts solid copper 2 oxide with dilute sulfuric acid to prepare a solution containing copper sulfate only. Describe clearly a method for this preparation. So we're trying to prepare copper sulfate solution from a solid. So of course we're trying to explain neutralization method. So what is the first thing we should do? We should add solid copper oxide using a spatula to 25 centimeter cubed of warm sulfuric acid in a beaker until excess solid remains in the beaker. You can also mention that the 25 centimeter cubed of sulfuric acid was measured using a measuring cylinder. In all of these, it's a very good idea to mention all apparatus. Then what should we do after that? Filter through filter paper and funnel. Of course, what is collected as residue is the excess copper oxide. What is required is a solution. So we can stop here and say that the filtrate is the solution. The question did not ask for crystals. Of course, if it asks for pure dry crystals, then we should go on and continue and say heat to point of crystallization, cool, filter the crystals. Then another type of questions has 7 marks on it, and here we need to explain in detail. So, for example, a student has clothes made of 3 different materials, cotton, polyester, and wool. After washing the clothes, the student observes the wet clothes becoming dry as the water evaporates. The student wants to know if the material of the cloths affects the rate at which the water evaporates. Plan an investigation to determine what? To determine how the material of the cloths affects the rate of evaporation of water from the clothes. You are provided with samples of cotton, polyester, wool, water, and you may use any common laboratory apparatus. Include in your plan, and please pay attention to what he says you should include in your plan. The apparatus, so you need to mention all apparatus needed. A brief description of the method, explaining any safety precautions. Do not forget to mention safety precautions, what you will measure, which variables you will keep constant, how you will process your results to draw a conclusion. You may include a label diagram and you may include a results table. You don't need to put any readings in the table. So the first thing we're going to ask ourselves, we're trying to plan investigation to determine what? How the material, so we're changing the materials, cotton, polyester, and wool, how the material affects the rate of evaporation of the water. So the first thing we need to do is, what are we going to cut samples of each material to equal sizes to show that this is kept the same for all the materials. A safety precaution could be wear gloves to avoid skin cuts. Put each material into a beaker of water for the same amount of time, for example 5 minutes. Remove and weigh using a balance. So this is the initial mass with the water, and then spread each piece and put in the sun somewhere for 1 hour, so same time, at the same temperature. Determine the time using a stopwatch, and then we left it in the sun for 1 hour, and then we're going to reweigh all of them, determine the change in mass, repeat each experiment twice and calculate the average change in mass for each type of material. Remember that readings should always be made twice and you obtain the average. The one with the highest decrease in mass has the fastest rate of evaporation. And then you could draw a table that looks like this, in which we have for each material, we have trial 1 and trial 2, and then what are we measuring? The initial mass, the final mass, the change in mass, and the average. And of course, you don't put any numbers into the table, you just show what the headings and the units actually should be. Okay, another experiment. Photosynthesis takes place in aquatic plants that live in water. Photosynthesis produces bubbles of oxygen gas. The greater the rate of photosynthesis, the faster the oxygen is produced. This figure shows apparatus used to investigate the effect of light intensity on the rate of photosynthesis. Plan an investigation to find the relationship between light intensity and the rate of photosynthesis. You are provided with the apparatus, and you may also use any other common laboratory apparatus. In your plan, you should include what? Any additional apparatus, description of the method, any safety precautions, what you will measure, which variables you will keep constant, how you will process your results and draw a conclusion. You may include a table that can be used to record the results. Okay, so we're trying to find the effect of light intensity on the rate. So what can we do? We put the lamp at a certain distance from the beaker, measure the distance using a ruler, record the distance in a table. Safety precaution, wear gloves, do not touch the hot lamp to avoid burning the skin. Remember, when you mention a safety precaution, you should say why we're doing that. Count, what are we going to measure? We're going to count the number of bubbles of gas collected in 5 minutes using a stopwatch. So in a specific time using a stopwatch. And then what do we want? We want to change light intensity, so we're going to repeat at different distances from the plant using the same what? The same lamp, so that we're not changing the lamp, same plant, same temperature of water, and we're counting number of bubbles in the same time. And then we can say repeat each distance twice or 3 times and take the average. Record the results in a table of what? Of the distance in this unit, centimeter, versus the number of bubbles. So the table can look something like this: distance in centimeter and the number of bubbles for the first trial and second trial, and then the average for each of the distances. Okay, now another experiment says magnesium carbonate is a white solid that decomposes when heated, so it gives carbon dioxide plus magnesium oxide. Plan an investigation to find the relationship between the mass of magnesium carbonate and the volume of carbon dioxide made. You are provided with magnesium carbonate powder. You may use any common laboratory apparatus. In your plan, include the apparatus, a brief description of the method, safety precautions, what you will measure, results table to record the measurements, how you will process your results. You're not required to include any results in your table. You may include a label diagram. So this could be the apparatus we're using, a test tube connected to a gas syringe or a flask connected to a gas syringe. Weigh 2 grams of powdered magnesium carbonate using a balance. We mentioned the apparatus. Put into a boiling tube connected to a gas syringe. And then what should we do? Heat the tube with a Bunsen burner. Determine the maximum. This is what we're measuring, the maximum volume of gas collected. Repeat using the same mass. Take average results to avoid anomalous results. So that's something we should say. Safety precautions: wear eye goggles to protect the eyes from the powder. Hold the test tube with tongues to avoid burning the skin. Then we're going to say repeat using what? We're going to repeat using different masses of the carbonate and determine the maximum volume of gas collected in each case. Draw a table of what? Of the average results, and draw a graph of the mass in grams versus the volume of the gas in centimeter cubed. You always mention units. Then you can say that a straight line curve indicates that as the mass increases, the volume of the gas increases. Okay, another question: when a person exercises, their pulse rate changes. Plan an investigation to determine the relationship between intensity of exercise and change in the pulse rate. In your plan, include the apparatus, description of methods, safety precautions, what you will measure, which variables you will keep constant, how you will process your results. You may include table. You don't need to include any results in the table. So let's see. So we're trying to determine the effect of intensity of the exercise on the pulse rate. So for a student, we measure the initial number of pulses, for example, in 15 seconds. Allow the student to do a certain exercise. So I chose walk on a treadmill for 2 minutes at a certain speed. Measure the number of pulses in 15 seconds when he finishes the exercise. Now determine the difference between the initial and final pulse rate. Allow the pulse rate to return to normal before doing the next one. The student then walks for 2 minutes at a faster speed, for example. Measure the pulse rate. Determine the difference between the initial and final pulse rate. Repeat at increasing speed, measuring the initial and the final. Repeat with other students. So this is just for one student. Now repeat with other students, same age, gender, height, weight. For each student, draw a curve of speed of walking versus the pulse rate. Okay, and then we can draw also a graph. Okay, another type of experiment. Urease is an enzyme that breaks down urea in solution to form aqueous ammonia. Urea in solution is almost neutral. The aqueous ammonia formed increases the pH of the solution. So we're being told that when we add urease, the pH of the solution should increase due to the formation of ammonia. Now, plan an investigation to determine the relationship between concentration of urease and pH of the solution formed. We are provided with urea solution, 10% of the enzyme, which is the urease, universal indicator solution, pH color chart. You may use any common laboratory apparatus, include the apparatus, the method, the safety precautions, what you will measure, which variables will be kept constant, and how you will process the results. So for an experiment like this, for example, I can say put 20 centimeter cubed, the specific amount of urea solution, into 5 test tubes. Usually we do at least 5 different readings. Using a measuring cylinder, add 3 drops universal indicator solution to each test tube. put the test tubes in a water bath at the same temperature. So this is an enzyme reaction. It would be best at something like 30 degrees Celsius. Safety precaution, wear gloves to protect the skin from the heat. Then what are we going to do? To the first test tube, add 20 centimeter cubed of the enzyme, urease. Leave for a certain amount of time. Compare the color of the solution. Remember, the test tubes have universal indicator and we have a chart that we can compare to. Record the pH. Then to the second test tube, we add more of urease, but remember that you want the total volume to be the same, so that the change in volume does not affect the color that you get. So if we have 20 centimeter cubed of urea, for the first test tube, we put 20 centimeter cubed of enzyme. Now, second test tube, we're going to change. So we're going to put, for example, 18 of enzyme plus 2 of water. Repeat using, for example, 15 of enzyme, 5 water. 10 enzyme, 10 water. So that the total volume is always the same. Record the results in a table. Repeat each reading twice to identify anomalous results and take the average. Draw a graph of what? Of volume of urease versus pH, and you have something like this, in which we have the different experiments, volume of urease, volume of water, and the pHs at each trial and the average pH. Okay. Another question. Citric acid reacts with aqueous sodium hydroxide as shown in the word equation. In the reaction, aqueous sodium hydroxide is neutralized by solid citric acid. So we have sodium hydroxide is an alkali, a solution. When citric acid is added, the pH decreases. Now plan an investigation to determine what? The relationship between mass of citric acid added to the aqueous sodium hydroxide and the pH obtained. You are provided with solid citric acid, aqueous sodium hydroxide, universal indicator in a pH chart, and the same types of instructions. So what should we do here? We have again a certain amount of the sodium hydroxide in 5 test tubes using measuring cylinder. Wear gloves to protect the skin. You remember the base is corrosive. Then we're going to add 3 drops universal indicator solution to each test tube, compare the color to the chart, and record the initial pH. So of course the initial pH would be something very high. This is an alkaline, strong alkali. Then we're going to start adding citric acid. So weigh, for example, 10 grams of citric acid using balance, add to the first test tube, leave so that it can react for about 2 minutes, and then compare the color to the chart, determine the pH. Repeat using different masses of citric acid. Usually, again, we want 5 different masses using the same what? Same volume of sodium hydroxide, same concentration of sodium hydroxide, and same temperature. Then we need to repeat each reading twice, take the average, record the results in a table, and draw a graph of the mass in grams versus pH. So your table could be something like this. Are we following how to think about all of? Okay, then this is another question. Plan an investigation to find out how much energy is required to increase the temperature of different liquids. So we're provided with 4 different liquids: water, salt solution, vegetable oil, vinegar, an electric heater, power supply, and we have this apparatus. And we know that the energy is equal to VIT. So in your plan, we're going to mention the method, what we're going to measure, and so on, the same instructions. So what can we do? We want to determine the different materials need different amounts of energy. So what we're going to do is we're going to put, for example, a specific amount of the first liquid. So 50 centimeter cubed of the first liquid into the beaker using a measuring cylinder. Measure the initial temperature using a thermometer. Record in a table. Then we're going to connect to the circuit, close the switch, start a stopwatch. And we will record the readings on voltmeter and ammeter, heat the liquid till the temperature increases by a certain amount. So either we heat it for a certain amount of time or we heat it till the temperature increases by a certain amount. Determine the time needed using the stopwatch, record in the table, and we use these results to calculate E. Of course, you're going to repeat using 50 centimeter cubes, so the same amount of the other liquids, same initial temperature, same voltage, same current, and we're going to record the time needed for the temperature to increase by the same amount. Calculate E using the equation, record all readings in a table that can look like this. And of course, the readings, you can repeat each reading twice to identify anomalous results or to take average, which is more accurate. Compare the values of E to determine which one is the greatest amount of energy. Okay. Then, another question. A student rolls a toy car down a ramp of height h. At the bottom of the ramp, the car rolls a distance d along the horizontal floor until it stops. Plan an investigation to determine the relationship between the height of the ramp and the stopping distance, which is the distance the car rolls along the floor. You are provided with a ramp that can be adjusted, toy cars, and basically the same instructions. So what can we do? We want to see the relationship between height and the distance the car will need in order to stop. So we're going to put the ramp, for example, at a certain height at the beginning. Put the car at the top of the ramp, release it, allow the car to stop, and measure the distance using a ruler. Then you're going to repeat using the same what? The same car, the same ramp, but at different heights, for example, 20, 40, 60, 80, 100, and so on. Record the stopping distance in a table, and we can repeat each height twice and take the average to obtain more accurate results, avoid anomalous results, and then we will need to plot a graph of height in centimeter versus distance in centimeter. So your table could look something like this. Are we okay with all of this? Okay, another one. Cetyl alcohol is a white solid with a melting point of this. If salt is added, the melting point decreases. Some of the apparatus needed to determine the melting point is shown. So you have a large test tube in a beaker containing water, and you're supposed to put the cetyl alcohol in that large test tube. Heat the beaker of water using a Bunsen burner. We're required to plan an investigation to find what? The relationship between the mass of salt added. So every time we have powdered cetyl alcohol, we're going to add a certain amount of salt, and we want the relationship between the mass of the salt and the decrease in the melting point of the cetyl alcohol. You're provided with powdered cetyl alcohol, powdered salt, and the apparatus that we had, and basically the same instructions. So what should we do? Weigh 20 grams of the cetyl alcohol. It's in powder, so this is going to be in grams using a balance. Keep the alcohol, this is a safety precaution, keep the alcohol away from open flame. Put into the test tube containing a thermometer. Add 5 grams of salt, so this is a certain amount of salt. Put the test tube into a beaker of water, heat the beaker, and stir the solid until it melts. Remember, when we're determining the melting point, we do not measure the temperature at which it melts. We're going to melt it and then leave it to solidify. So we take the temperature at which it becomes a solid, which should be the same as the one it melts. So for safety, wear gloves to protect the skin from the hot test tube. remove the test tube when the mixture has melted, stare with the thermometer until the liquid in the test tube solidifies, and that is the temperature we're going to record. Again, another safety precaution could be wear safety goggles to protect the eye from splashes. Repeat using different amount, the same amount of citric acid but different masses of salt. So we could suggest 10, 20, 40, whatever of salt. Repeat each reading twice. Take the average to obtain more accurate results. Avoid anomalous results. Draw a graph of the mass of the salt versus the temperature. And this would be an example of the table that we can use. Getting familiar with all of this? Okay. So that is one type of questions. Another type of questions that you get in paper 6, of course, are the tests. So I just chose some of the common tests that are being asked. So for example, this was a question where it said, when the student tests the gas with a lighted splint, there is a squeaky pop. Identify the gas. Of course, if we're testing a gas with a lighted splint, and it gives a squeaky pop, then the gas is hydrogen. You have to know that. Another type of question where he gives the results of the tests. So he's saying we have tests with dilute nitric acid and barium nitrate, and it gave white precipitate. Of course, you should know that nitric acid and barium nitrate is a test for sulfate. So when the question says state the name of the anion, it is sulfate. Now, the other test that he had was excess sodium hydroxide, and he got green precipitate insoluble in excess. This is test for what? Which one gives a green precipitate with sodium hydroxide? Iron 2, and you have to specify 2, because if it's iron 3, it would give a reddish brown precipitate. So you have to identify that this is iron 2. Another kind of question is where you're required to take a reading from what he got. So, for example, when we're reading measuring cylinders, please remember we're reading measuring cylinders, so we're reading from the bottom. So this is what? So this is, for example, the first one is 41.0. Remember, if one of the readings is to 1 decimal, all readings must be to 1 decimal. So the second reading here is what? It's between 18 and 19, so it's 18.5. Remember, you're looking at the bottom of the meniscus. So that means that first one has to be 41.0, not just 41. Another kind of question is where you're required to read the time from a stopwatch. So, for example, he says record the reaction time to the nearest second. So remember, nearest second. What you have in that first stopwatch on the left is 30.09. So to the nearest second, that would be 30. But the other one on the right is 30.69. So if we're reading it to the nearest second, it is 31. Here, for example, he's saying record to the nearest second. So here we have 1 minute and 27.87 seconds. That's what you have in that stopwatch. So how do I record it in seconds? Of course, the 1 minute is 60 seconds plus the 27.87 is actually 28. So the total seconds is 88 seconds. I hope you're familiar with all of this. This is a thermometer. What do we read from this thermometer? This is, of course, something in between. So this is going to be something 0.5. So this is obviously 24.5 degrees Celsius. When we're reading a voltmeter on a meter, remember that the voltmeter here, for example, is from 2 to 3. So that means each small line is 0.1. So that voltmeter on the left is what? It's 2.6. What about the ammeter? The ammeter is going 0, 0.2, 0.4, 0.6, 0.8. So that arrow is pointing to where? That is actually 0.25. This is in the middle between 0.2 and 0.3. So this is 0.2. Okay, another test is describe a test to confirm that the gas is oxygen. What is the test for oxygen gas? You should be familiar with the tests for oxygen gas is a glowing splint. Insert a glowing splint, it relights. And that's the end of this review. I hope this was useful to you. Please make sure you understand all of this, and good luck in your exams.